maybe 10 years ago you visited Taiwan.
I think it was more than that. I think it was in 2005. Oh no. Yes, I think it was in 2005. I think it was a very long. Yeah,
I
very
still
long
remember
time.
your daughter.
Yeah, that's right. She was. I have pictures of her. I've just got a book and I've got a picture of that. One moment, I'll show you.
Okay, good.
Hi, 张老师。
Hi, 姚医师。
Hello.
There. Can you see?
Wow. So grown.
Yeah. That's when we were there.
Wow. Good to see her.
Yeah. No. She's 22 now.
Oh my goodness.
Yeah. Yes.
Well, thank you so much for accepting the invitation.
Well,
yeah.
I'm delighted to be here. Yes.
Okay.
So there's going to be a 5-minute introduction before my talk. Is that right?
Yep. Just really brief introduction, so I save most of the time for you. I think most of them just want to hear your lecture.
Okay.
So this is Dr. Liu Yiqian. You can see from screen.
A moment, he's disappeared off.
Hi, Professor.
Oh, okay.
Hello.
Hello. Excuse
Hi.
me a moment. I can't see you. I'm not quite sure what's happened.
Oh.
No, I appear to have lost the whole page. That's strange. It's asking me to do something. I'm going to have to come back in again. I'm sorry. You've disappeared entirely. I can hear you, but see if
maybe join us again.
Okay, all right. I'll join again.
Okay.
Oh, okay. I see. My apologies. My apologies. It didn't go off. It's just has a different sign. Okay, Amora.
Okay, good.
Oh, good.
So, should we get started?
Yeah, sure. Of course.
Okay. Good afternoon, ladies and gentlemen. Welcome to the Frontiers in Dementia Research, the Master Lecture. I'm Tylee from Academia Sinica, and it's my great pleasure to welcome and to introduce today's speaker, Dr. Cathy Price. Dr. Cathy Price is a professor of cognitive neuroscience at University College London, and she's also the director of the Wellcome Trust Center for Neuroimaging. Dr. Price is a world-leading scientist in using neuroimaging technique to study language processing in the brain, and her remarkable research has greatly advanced our understanding of how language process interprets with other cognitive function in health and diseases. And her recent work is to develop a database so that we can use the database as a tool to predict patients' language outcome and recovery after stroke. So with those important contributions, Dr. Price received the Savage Award for Life Science in 2018. And recently, she also has been elected as a Fellow of British Academy and a Fellow of the Royal Society. So I can go on and on for those awards she has received for maybe 10 more minutes, but I think we all can wait to listen to today's lecture. So without further ado, please join me in welcoming Dr. Price.
Thank you very much. I'm just going to share my screen. I'm delighted to be with you. It's very early in the morning for me, but I have great memories of my trip to Taipei in 2005. So I can imagine where you are, and I wish I could see your faces and meet you. But anyway, I'm going to share some of the work that I do in a talk. And what I'm going to do is to talk about language in the neurotypical brain and how we might use this knowledge to predict the outcomes and recovery for people who have brain damage, particularly those patients after stroke. I've arranged, I've arranged my talk into 4 main sections. The first section will focus on the neurotypical brain and just a reminder of the brain regions that are involved in naming, reading, and repetition. Because these are the sort of key core tasks that underlie our ability to generate language. I'm then going to look at the effect of damage to some of these regions and how that changes object naming and reading ability. And part of that will show you that for some regions that are crucially involved or consistently involved in language in the neurotypical brain, patients can have recovery afterwards. So they might be impaired and they may never have an impairment after removing part of the language system, or they might have a good recovery such that they can read and name or repeat again. And so the third section of my talk will discuss how this might occur via functional reorganization after damage to the normal system. And finally, I'll think about the recovery pathways and how they might emerge from normal variability of function within the neurotypical brain itself, that our brain is capable of doing the same thing in many ways. And this is something that I refer to as degeneracy. And then finally, just a little bit on future directions. So moving to the first section. Slides on. So the first section, apologize for the error there. This is supposed to be functional, not functional. Functional anatomy of naming, reading, and speech in the neurotypical brain. I'm going to discuss one fMRI experiment, which is in neurotypical controls. And the goal of this experiment is to segregate the neural systems that are associated with auditory and visual processing, so sensory processing, semantic identification, that's recognizing an object or a word that you hear or see. Inner speech, that's when we hear that's the sound of the words in our head. Speech production, that's when we produce those words. And then executive functions, which include a whole collection of skills, including our ability to attend, to remember what's there, to manipulate things in our head. So these are higher order functions that are involved in many tasks that we perform, movement as well, but including language. And of course, as you will know, functional MRI, that we're going to use fMRI, infers neuronal activity related to functions of interest by measuring changes in blood flow and oxygenation across different tasks and conditions. And we want to know which brain areas are activated in our tasks of interest, in this case, language tasks, but we can't identify what those regions are unless we have a very good control tasks. And these are baseline tasks that are controlling for parts of the task that we're not interested in. So before I even show you the results, I need to show you the tasks that I've asked them to do and the baseline tasks critically. So in the task for this fMRI experiment, they had two different tasks. One was speech production and one was a hand response, just responding with two fingers. And they had three types of stimuli. They either saw pictures of objects, written objects, so that's text, or heard objects, and so that's speech. And the speech production tasks then were naming the object, reading aloud, and repeating aloud. Those were the speech production tasks. And then the hand response was always a one-back matching condition on exactly the same item. So everything was the same except for the final response. And then we can hypothesize what are the differences between the different tasks that we're using. So obviously, when you're looking at pictures of objects, you've got to recognize the visual form. When you're looking at written object names, you need to recognize the orthography. When you're hearing, you've got to have hearing object names, you've got to use auditory processing. And there's going to be common language between them all. They're all going to involve some sort of semantic identification in order to produce the speech. And in addition to that, written object names and heard object names, we hear the sounds in our heads, the phonology of the words. So we can hear speech in our heads. When we're looking at pictures of objects, we don't go around the world generating object names. And there's lots of evidence to show how we will be generating in a speech much more when we're reading than when we're looking at our normal visual world. But for pictures of objects, we will generate in a speech when we're asked to name them. So if I'm asked to name a cup, I will generate the sounds in my head before I produce the word cup. Finally, we also have these executive functions, and these are the same across all the tasks, but to varying degrees. So a task like object naming requires more attention and cognitive control, because you've got to choose which name you're going to give the object. And some objects have multiple names that we have to decide, and which one we're going to say and which one the experimenter wants us to say. Whereas when you're hearing object names and you're repeating them back, it's a fairly simple task. You hear them and you don't have to decide what word to say because you've already been told what word to say. So whilst all these executive functions are involved in all three tasks, they'll be there to different degrees, and we have to take that into account. So I'm going to start by looking just at the brain regions involved in visual orthography versus hearing the words. So this is comparing reading written object names with repeating heard object names. And as expected, what you see is if you're hearing the speech and repeating it, auditory repetition increases activation in the auditory cortices. These are the areas in blue. Here you can see it's around Heschel's gyrus. Oh, my pointer's not working. There we go. Around Heschel's gyrus here. And in the if you reverse that contrast and you say, well, what areas are more active for reading than for auditory repetition, then we see these red areas here that are involved in visual and orthographic processing. And you can see that the activation extends along the occipital temporal sulcus here, particularly strong on the left. And this region here has often been referred to as the visual word form area. Although I believe it's involved in visual word form processing, the key point that I've made multiple times is that this area is not specific to orthographic tasks. It's important for reading, but activation here is even higher when you ask people to name the object than to read it. And interestingly, if you ask them to do tasks which don't involve naming, the activation isn't as high. So I can come on to discuss that if anybody's interested. But the key point here is that this is a region that's important for reading, but it's not specific. It's not only involved in reading, it's also involved in naming objects. So it has a higher function. And we can split this region into two parts. We can split it into the posterior part here, this visual part, which is more activated for object naming than reading, than fixation, but it's not involved in auditory repetition, which means that it's more visual in its response. Whereas the part in front of that is also involved in auditory repetition more than fixation. So this part here is more involved in the semantic, the anterior part is more involved in the semantic identification of items, and it's most activated for the visual stimuli, but it is still activated for auditory repetition. And we can discuss, why this might be the case, whether it's actually at the semantic identification level. Some people have argued, well, maybe people imagine what a word looks like when they're saying it. But I don't think the evidence is strong for that, but it is one hypothesis that people can generate images in this region. And I'm now going to compare these activations here from fMRI with a model from 1986 that Luders produced, looking at brain stimulation work. So this was electrical stimulation in patients who were being operated on. And this electrical stimulation to this basal temporal language region down here, the area I've just been talking about, the yellow area. If you stimulate this region, Luders reported receptive and expressive aphasia, including difficulty with auditory repetition, difficulty with naming, which is called anomia, difficulty with reading, which is called alexia or dyslexia, and impaired writing, difficulty with agraphia. So, the fMRI results are consistent with its electrical stimulation work. So coming back to our fMRI study with neurotypical controls, the next areas I'm going to show you are those involved in inner speech. So this sometimes, or you could call it phonology, but this is hearing, generating the speech in your head, but without saying it. And this can happen very automatically when you're reading. And the argument would be that inner speech is involved in all 6 of these tasks, with the exception of when people are doing one-back matching on objects. So I should have explained or reminded you that a one-back matching task is when you have objects that are presented to you, and you're not asked to name them, you're asked to say, have I just seen the same object again? So they'll see a series of objects, and then there'll be, sometimes the objects will change, and other times they won't change, and they have to indicate when did I see the same one. There was a repeat, though I saw the same one as in the previous presentation. So they don't have to name it to do it, because they can see visually that there's been no change in the stimulus. And so this task doesn't require you to name them. In fact, if you were naming them, you'd be very slow and it would be very inefficient. So one moment, someone's just asking if they can annotate the shared content. I'm going to approve that. Is that okay for me to approve that? Okay. All right. Sorry, I've been distracted because I had a sign requesting me to approve. I've just lost my mouse button a moment. Oops. Now I'm drawing on my slide. I didn't want to do that. I do apologize. I'm having a little technical issue here. I need to work out how to turn that off. Oh, I do apologize. Let me see if I do apologize. My slides have stuck. See if I can I had a message come up on the screen asking permission to annotate. I thought it was coming from externally and I agreed. And it's now blocked my slides, and I don't know if you can see yellow marks came up on it, and I've lost my cursor. Would it be possible for you to disconnect me? Would it be possible for you to disconnect me and I will come back straight back in again?
不好意思，请稍待一下，我们让他再登录看看。
江老师，嗨，我们先让他出去，然后再进来，这样。So, Dr. Price, is this good now?
Yes, this is good. I've come back again and it's
Okay,
now
good.
Okay. Can you see my slides again? I apologize. I apologize for that. So now we're looking at the brain regions involved in inner speech. You can hear me okay, yes?
Yes, we can hear it very clear.
Okay. Good, thank you. So when we look at the areas involved in inner speech, what we see is something that should be recognizable to most people familiar with the classic functional anatomical model of language, which is that we see activation firstly in the posterior temporal sulcus on the temporoparietal junction, which is sort of where you would expect Wernicke's area to be. Wernicke's area is associated with auditory images of speech in the classic neurological model. And we also see activation in the ventral part of the inferior frontal cortex. So not quite where we expect Broca's area, but an inferior part of Broca's area. So we have some frontal and temporal regions that are associated with inner speech. Again, as I said with orthography, these areas, though, are not specific to the sounds being speech, because they're also activated when we ask people to listen to non-verbal sounds, so environmental sounds which don't involve any speech and don't require any association with speech. So the argument might be here that these areas are involved in auditory short-term memory, And auditory short-term memory is what we need when we're speaking to hold the sounds, the images of the sounds in our head. And if I now compare these regions, though, to the neurological model of language from electrical stimulation studies, you can see the similarity between the results of the electrical stimulation studies and the results of the fMRI. So these top regions here associated with inner speech in yellow, you can see here the ventral part of Broca's area here and the Wernicke's area. And then as I've already shown you, the basal temporal language area here being involved in semantic identification here. So the fMRI is consistent with these electrical stimulation studies for language regions. I'm now going to go on and show you the executive functions. So for the executive functions, as I described before, these are involved in all the tasks. They may be differentially involved in object naming relative to heard object names, but we still expect them to be involved in all tasks. So we can ask what's common to all 6 sensory tasks. Now, when we do this, what we see is that the pars opercularis, which is very commonly referred to as Broca's area here, is activated for all 6 of these tasks, not just the speech ones, but even when they're doing these one-back decisions on pictures of objects. We also see activation in the inferior frontal sulcus and the dorsal premotor area, which is sometimes referred to as area 55B. In the parietal, you can see the supramarginal gyrus here. And then the SMA and the pre-SMA and the anterior cingulate, see, which are common, well, the anterior cingulate is a common executive region involved in attentional control. And then bilateral anterior insulae that you can see here, and bilateral cerebellum. So here, though, the key thing here is that the area that people classically associated with speech production, Broca's area, is involved in all of the tasks and is more likely to be domain general executive function than speech production. So now if we talk about the areas involved in speech production, here we expect those to be common for object naming, reading aloud, and repetition, and more active for speech production, the hand response, because the hand response doesn't involve any speech production. And these are the areas in green here of the brain regions that are more activated for speech production than the hand response. And as you can see, we see these motor regions here involved in the control of speech, or the control of your larynx in particular, and your breathing, and moving the mouth and face. And these are the motor regions. You see more on the anterior cingulate, and we see these activations at the back part of the putamen bilaterally, posterior part of the putamen bilaterally, and in the cerebelli, as you can see here. So in summary of that experiment, I've identified 4 systems, and showing the mid-fusiform is involved in semantic identification. The posterior superior temporal and frontal opercular are involved in auditory working memory. These are our hypotheses, tasks that involve auditory working memory. The motor cortex, medial cerebellum, putamen, SMA, pre-SMA, and anterior cingulate, all involved in speech production. And then a set of regions, including Broca's area, that are in pars opercularis, that are involved in executive functions. But in all the work that I've done in 30 years, if you involve different sorts of picture tasks, we never see anything that is specific to orthography or inner speech. And so my hypothesis has always been that there aren't language-specific regions in the brain, but that language involves the coordination of multiple different regions that have different tasks and are brought together for the unique function of language. But of course, that's what I've shown you here is just at the single word level, and people can argue that it's different when you get to the sentence and higher communication level. Although again, there are no regions that we know of that is specific to syntactic processing in other studies. I'm now going to move on to section 2, which is what happens when we damage these regions. And I'm going to focus on damage to 3 different regions. Firstly, looking at the occipital temporal regions associated with visual recognition and semantic identification. Then at damage to the frontal regions involved in inner speech and executive functions. Then damage to the left and right putamen involved in speech production. And then finally, I'm going to look at the lesions that cause persistently impaired speech production. So this is just a single case, but there are many examples like this, where there's damage to the left occipital temporal cortex associated with visual recognition and the basal language area, and we know that it's important for reading. And these patients almost consistently have severe difficulty reading long words, but they have less severe difficulty with their writing and their object naming, and very often they preserve the ability to read short words. So I'm going to come back to this in a future session to say, how is it that they're able to read when they have damage to areas that we know are important for reading and object naming? But the point here is to show the consistency between the functional imaging data and the brain damage region. So you remove this region and they have difficulty reading, and they almost invariably have difficulty naming objects as well. Now, here are some examples of patients who have got extensive frontal lobe damage and damage to regions that we know are involved in inner speech and also executive functions. And the key thing about these patients is they tend to recover their speech production, providing the anterior part of the articular fasciculus is preserved. So this is the anterior part of the superior longitudinal fasciculus. So I've highlighted that in red here, approximately where it is. This is more on the surface of the brain. But when this region is preserved, if you damage the frontal lobe, then the majority of our patients will recover their speech production, despite having damaged areas involved in inner speech and executive function. So again, we have to come back and ask, how is this possible that they've recovered? Because it's not consistent with functional imaging literature. It's also not consistent with what we know from Broca's area, and I'll come back to that in a moment. So this here is an experiment. It's a lesion identification experiment that asks, well, which frontal lobe regions are important for object naming and reading? And here what we did was to take 134 stroke survivors who all had damage to the left frontal lobe, and to look at the degree of damage to different parts of the frontal lobe and surrounding areas. So the green region here is damaged to Brodmann's area 45. The red area is Brodmann's 44, and both these regions here are classically thought of as Broca's area. The yellow region is the ventral premotor cortex. The cyan light blue area here is the motor cortex. The pink is the insula, you can see here. And then black is the putamen, which you can see down in the bottom row here. And then these are the white matter tracts that we looked at. There's the anterior part of the arcuate fasciculus here, the unculate fasciculus, and the aslan tract, the frontal aslan tract, which you can see here at the bottom here, that runs from the motor cortex up to the SMA and the pre-SMA. So the experiment looked at the degree of damage to each of these regions and asked, where does damage, is it most consistently associated with speech production difficulties? And we found that the best predictor was damage to this anterior part of the arcuate fasciculus here. And when you accounted for damage to this region, there was no contribution, no additional contribution for damage to Broca's area in BA 44 and 45. So it's the white matter tract as opposed to the cortical regions classically associated with Broca's area that were critical for speech production. So this is where we get these regions here. When these are preserved, all of this damage here does not result in long-term speech production difficulties. So we can go back now to Broca's area and the work of Broca, who was the one who associated these frontal regions here to speech production. Nina Dronkers in 2007 did some brain scans of the brains that Broca preserved very sensibly 150 years ago, and because he never dissected them because he thought that they would be useful in future, so he looked at the surface of the brain, and from the surface of the brain you can see that the inferior frontal gyrus is damaged. But when Nina Dronkers looked at the brains, what it showed was that on the left side of the brain here, you can see that the damage extended all the way down the white matter tract in the arcuate fasciculus here. So you can see it's black here and damaged on the left, but on the right you can see that it's not damaged. So we now know that Broca's brains actually had this extensive damage, and from the lesion studies that we've done more recently, we know that we hypothesize that it's the damage to the white matter, not the damage to the inferior frontal cortex, that is associated with speech production difficulties. Now moving on to the damage to the putamen, we have a very interesting response with damage to the putamen, and I'm showing you here behavioral responses for patients with damage to the left putamen. For example, you can see one of the patients here, his damage to the left putamen, not the right putamen. And another example of a patient with damage to the right putamen, you can see it damaged here, but the left putamen is intact. And we have 24 patients with damage to the left putamen and 13 with damage to the right putamen. And we can look at their speech at different time points. So after damage to the right putamen, the speech is not very impaired at 1 week. It might just be slightly down, and then it goes up to normal or almost normal at by 1 year. But if we look at damage to the left putamen, this blue line here, at 1 week, damage to the left putamen will impair speech, consistent with fMRI studies showing that the left putamen is involved in speech production. But over time, we see by a year, these patients have recovered back into the normal range. So this lesion result is consistent with the fMRI at the beginning, the initial effect of damage, but the recovery shows the brain can recover from damage to the putamen, and or one or other putamen. And it's again a question we want to understand: how does the brain do that? Finally, in this section, these are the areas where we see the most consistent effect on speech production. So if you have damage to either this green area, which is part of the anterior part of the arcuate fasciculus underlying the motor cortex, or to this yellow area, which is the white matter underlying the temporal lobe in speech processing regions, if you have damage to either of these regions, there's speech production impairments. The incidence of speech production impairments is very high. So 97% of our cases with damage to the anterior arcuate fasciculus, 91% of our patients with damage to the temporoparietal white matter. So these regions are the most critical, and they do look in the vicinity of Broca's and Wernicke's area, but they're in the white matter underneath the cortical regions that are normally talked about. And finally, what this slide shows, that what we found is that if you preserve these regions, you preserve the superior longitudinal fasciculus, including the arcuate fasciculus and the temporoparietal component of it, as well as the left putamen and the white matter surrounding the left putamen. If you preserve these regions, then patients who are aphasic with damage outside these regions will recover. And this is where we get to what is useful clinically. What's useful clinically is that if these regions are preserved, we see very consistent recovery in patients who have damage that preserve these regions. And this is one of the findings that I want to write up as a potential clinically useful application. So the third section of the talk is to consider, well, how is it that people do manage to recover? What sort of functional reorganization is happening in the brain after damage to the normal system? And I'm going to look at fMRI studies of patients who have damage to left Broca's area in the frontal cortex, the right posterior cerebellum, the right motor cortex, and the left motor cortex. And in each case, we're going to do fMRI of the patients with these lesions and ask, is activation upregulated in undamaged regions? Is it upregulated in the left hemisphere or the right hemisphere? Is it within the neurotypical system? So other language regions are activating to take on the loss of the lesioned regions, or are there new regions that are not involved in neurotypical participants? And finally, are the results that we see lesion site dependent? So the first experiment here is looking at patients who've got frontal damage, and you can see the patients here on the left. You can see the frontal lesions here, left frontal. So the lesions aren't all exactly the same, but we asked, is there anything similar about these patients where they increase activation in other regions? And what we see is increased activation in the right posterior cerebellum that you see here in right. So activation, this area is activated when neurotypical participants are producing speech. It's even more activated in patients who've got damage to the left frontal regions. And in addition, there is a weaker and smaller effect in the right pars opercularis, which might be considered like a homologue area working a little harder. Now, we don't know exactly how this is happening, so we're not really understanding the mechanisms here, but we are seeing that something is happening that is after the lesion that is changed the pattern of activation in other speech production regions. So I just showed you that damage to the left frontal lobe, we see at the bottom, increases activation in the right cerebellum. We can also ask, well, what happens if you damage the right posterior cerebellum? Is there then more activation in the left frontal cortex? And the answer to that was no. Rather than see activation in the left frontal cortex, we saw enhanced activation in the right premotor cortex. So the right posterior cerebellum is damaged, the right premotor area has increased its activation, potentially to compensate for the loss of the right cerebellum. But we don't know what the mechanisms are. We can then go on and say, well, what happens if you damage the right motor cortex? Then which areas you see? And here we see patients who've got damage, extensive damage in the right hemisphere, including the right motor cortex. And what we see in them is that they are increasing activation in the left posterior superior temporal sulcus here, which we associated with inner speech, and in another part of the cerebellum, the right cerebellar vermis, which you can see here. So you can see each time we damage a region, we're seeing different patterns of activation for these patients. And finally, in this one, we look at damage to the left motor cortex as opposed to the right motor cortex. And now we see damage in the subcortical regions here. In the yellow regions here, in subcortical regions, increasing after damage to the left motor cortex. So to summarize these findings, we do see lesion site dependent changes, and they're within the normal speech production system. So to answer the questions that I posed at the beginning, is activation upregulated in undamaged regions? And the answer is yes. Is it activated in parts of the neurotypical system? And the answer is yes. Is there increased activation in new regions, those that we don't see in the neurotypical brain? And the answer is not at the group level. Is the effect lesion site dependent? And the answer is yes, but we probably need to have larger sample sizes and compare these patients in large groups. So more work is needed to be clear about that. And is it in the left or the right hemisphere? And the answer is both, but it depends on the lesion. And then to summarize exactly what those effects were again, damage to Broca's area, increased activation in the right cerebellum. Damage to the right cerebellum, increased activation in the right motor cortex. Damage to the right motor cortex, increased activation in the left posterior superior temporal sulcus and the left cerebellar vermis. And damage to the left motor cortex, increased activation in the left caudate, thalamus and pre-SMA. So what could possibly be happening here, and we need to do more work on it, is it could be that every time you have damage to the system, the whole of the rest of the language system has to work extra hard. And each time we introduce new groups of patients, we may just be seeing different tips of the iceberg, which is why I keep arguing that we need to continue to replicate these findings to know, is it the whole language system working harder, or is it really that different parts of the system, they pair up in different subsystems that compensate for one another. So there's lots of work to be done there, but they're clues as to where we can go to try and understand this better. And finally, I want to talk about sort of theory behind this and recovery pathways from normal variability in function. I'm going to come back to this result looking at the putamen, damage to the left putamen, where we can see this recovery occurring over time here. and we can ask which brain regions might be compensating for this, and what happens in the normal brain. So we can investigate this using a technique known as dynamic causal modeling, where we look to see how activity in one part of the brain, for example, the occipital temporal region here, connects up to activity in other parts of the brain. So in this case, we're just going to show a model with 3 different regions: the occipital temporal region, which is the basal temporal language area, a precentral region, which is a motor output region, speech production region, and the putamen. And using neurotypical controls, so this is not using patients, ask how do they connect? How is the putamen involved, and how consistently is it involved in linking activation from the inputs to the outputs? And if you look at this graph here, each plot here shows a different control subject, and you can see that some subjects have got high connectivity in 2A, which is the route via the putamen here, and more than in the other route, which is not involving the putamen. And other subjects down here are more strongly using this route than this route. Now, this is just in neurotypical controls, but these sorts of results with neurotypical controls allow us to produce hypotheses, because one, it shows us that even in neurotypical controls, the putamen isn't always involved in producing speech. There are other routes that could be used. And this doesn't mean there's a direct white matter connection between the two. There'll be multiple other regions that will relay and be involved. It's an oversimplified model. But it means that there are other ways other than putamen in the neurotypical brain. And if we as individuals have a preference for using one processing route versus another, then when we have damage, our speed of recovery might depend on whether or not we've had damage to a processing pathway that we used to use a lot, but there are others available that we're not practiced using, in which case we have to practice using those other ones available. Or it might be the case that we have damage to a pathway that we never really used much before, and we can still use the same pathway that we did previously, in which case we may have very little effect on our speech production if we were to have damage to, say, the putamen here. So the point is that there are different, in the neurotypical brain, there are different routes available or different processing pathways, and that might be the organization that allows patients to recover. I'm going to show you another example of that, and that's in this patient that I showed earlier with a large occipital temporal lesion here in reading areas. And as expected, the patient got severe difficulty reading words, particularly long words, but is able to read these short 3 to 4 letter words. So we can ask, how are they reading 3 to 4 short letter words? And this is a slightly more complex model here, where we've got 5 different regions linking the occipital lobe via the ventral occipital cortex to the premotor and the motor regions, and either going via the occipital temporal cortex or going via the superior temporal sulcus. And what we see in the patient is that their connectivity, because they've got damage here, they show strong connectivity during the red regions when they're reading short words. So they're bypassing via connections that don't involve the occipital temporal cortex. But then we can look in control participants and say, well, are there any control participants that do the same when they're reading short words? And the answer there is yes. So again, neurotypical brains, we will vary in how much we engage the ventral occipital temporal cortex, because it's not needed for reading short words. It is needed for reading longer words, but it's not for reading short words. But this model helps us to explain how the recovery might be occurring. I will conclude there and thank the many people who have done the work over the last 30 years, which I'm summarizing the results from. I've got many people involved in the patient management and recruitment, and the data management, many people involved in the fMRI analysis and data acquisition, and many people involved in the data analysis. And I particularly like to mention Professor Mohamed Seguier, who's really been inspirational in both the conceptual and the analytical part of the work that I do. So thank you very much indeed.
Professor Price, thank you so much for your wonderful speech. Thank you.
Thank you.
Yes, I'm a clinical neurologist. I'm just very interested about the region you just mentioned. Is the anterior arcuate fasciculus.
Yes.
Yes. It's very interesting because DTI study in primary progressive aphasia (PPA) patient also have a very specific damage to this area. Yes, and however, I think we didn't see compensation in this group of patients. I mean, the non-fluent progressive aphasia patient. So I'm just wondering, I mean, is the compensation not happening in some patients or just other reasons?
Well, it depends on the extent of the damage.
Yes.
So the recovery is occurring if the anterior part of the arcuate fasciculus is preserved. If it's not preserved, then they won't recover. But we do have patients who, not very many, a small proportion of patients who do make a recovery and are able to speak after damage to the anterior part of the arcuate fasciculus. But generally it's years, so we're talking about 5 to 10 years later. But I think with primary progressive aphasia, it's difficult to say how extensive the damage is.
Yes.
Surely. And the disease is progressive, which might mean that the process of recovery is impaired by the progression of the disease, and so you don't see the compensation. Would that make sense of your data?
Yeah, maybe we have to, because we didn't do the fMRI on every primary progressive aphasia patient. Yeah, maybe we can find out later.
Yes.
Yes.
Well, I'd be very happy to discuss and put it into any of the context if that's helpful. We could maybe continue the discussion.
Yeah, yeah. I'm very happy to give our data. Yes. Another very interesting part for clinical neurologist, I think maybe you have noticed is a cross aphasia.
Yes.
For some patients, they have a right damage on the right hemisphere. However, they have a very severe dementia, severe aphasia.
Yes.
Yeah, it's very interesting. Yeah.
Yes, this is certainly the case. We see quite a lot of crossed aphasics. There's two things. One is there are some regions in the right hemisphere that are important for speech, and as I showed you with damage to the right motor cortex, but these patients had very extensive damage. We see increased activation in the left language regions. So just the fact that we see that, and we've observed that in patients who've had neurosurgery to the of the right hemisphere, as well as patients who've had strokes in the right hemisphere. The fact that the left hemisphere is working harder suggests that the right hemisphere was contributing. So it wasn't necessarily cross-aphasia. It's because the right hemisphere does contribute to our language, and it is important. And if we lose it, the left hemisphere has to work harder in order to compensate for it. So it has less effect generally on the behavior, but that if you really push patients into speeding up their responses and testing them at a final level, you will find a mild impairment in everybody. But then for some people, there are these, really aphasic responses, the cross-aphasia. Well, we have to assume that perhaps they were relying on their right hemisphere previously and had less involvement of the left hemisphere.
Yes, I found my patient has a cross-aphasia. They usually recover faster. I think their recovery is, yeah.
Oh, that's good to hear. That's good to hear, yes.
Yeah.
Yes. So they've an initial severe aphasia and then they recover.
Yes. Yeah, they recover rapidly. So I think we are going to take questions from the audience. Today's audience are mostly clinical neurologists, so sometimes they are very shy. No, yeah. So is there any questions from the audience?
There was a question that went into the chat, I think. I didn't read it, but I saw there was something in the chat. I'm wondering, do you find similar language change on white matter disease patients? Oh, it disappeared, the chat. One moment. White matter disease patients with lesions on similar locations. So I think the answer is generally in the patients that we have with white matter disease, it's very diffuse. There's not large holes in the brain, and it depends. That's the most variable, the effect of white matter disease. At some point, if it's bad enough, then we will see aphasic symptoms come. But for many patients with mild white matter disease, the effects are very inconsistent. So we do have patients who have, for example, speech production difficulties with damage to the mild damage to those white matter tracts. But generally, the impairments are seen for these patients who they've just lost them. They've been severed, these tracts. There's no, it's almost like a processing pathway, yes, that's been blocked. I hope that answers the question. If my questions, if my answers don't answer your question, I'm very happy to discuss by email later. There's a second question here. There's the same question. In caring of stroke patients, I've noted that some patients with left insular infarction also demonstrate various kinds of aphasia. However, the left, it's disappeared. However, the left insular does not seem to stand out in your analysis. What's your opinion on this? So I did show in the fMRI analysis that the insula was involved in all 6 tasks. So initially we used to think that it was specifically involved in speech production, but now our hypothesis is that it is involved in speech production, but because of executive type functions, not typical language functions. So it's important for speech production, but not only involved in speech production. It's involved in many tasks. I hope that answers your question. There's another question. I'm curious to hear your perspective on how left and right brain regions may contribute to language processing both before and after damage. For example, whether how these regions may collaborate or function independently to support language processing. Oh yes, so this is a fascinating issue. It's really about lateralization in the brain. I think when we are speaking, the two hemispheres are both contributing to language in different ways. So I would have to go back and show you it. Most of the fMRI activations I showed you were bilateral regions involved in speech production, bilateral regions involved, in executive functions. And when you have large lesions in either hemisphere, and people are speaking, we see the other hemisphere compensating, which again suggests to me that it generally uses both, because they're not working entirely independently of one another. The two functions are important, but they've clearly contribute in different ways. I hope that answers your question. That also the motor cortex, if you damage the left motor regions, we expected to see increased activation in right homologs, but we simply didn't find that. We saw more activation in the left hemisphere. So it's very difficult. I think we don't know the answers to these questions, except that both the left and the right hemisphere are contributing together. I don't see any other questions. I'm very happy to ask.
Perhaps the audience can send email to Professor Price later. Yes. So thank you everybody for joining the meeting, and particularly thank you for Professor Price. I'm very glad. I mean, very appreciate this very interesting talk. Thank you so much. I have so much questions I want to ask, but I think the time is very limited. So maybe later.
I look forward to hearing from you. Okay.
Thank you. Thank you.
Okay. All right. Okay. Goodbye, everybody.
Thank you, everybody. Thank you.
Have a lovely evening. My day's just starting.
Yeah. Okay.
Bye.
Bye-bye.